Hello everyone, welcome to the B.Pharm special program where we guide you to study better and in smart way. This is Swapnil Patil going to take one of the most interesting and learning topic of B.Pharm 5th semester on the important unit of enzyme 3, which is interesting unit of subject biochemistry. Are you ready then? Our topic is divided into the two modules, that is module 1, concept of vitamins as a coenzymes and their significance. Module 2, metals as a cofactors. Let's begin with module 1, that is vitamins as a coenzymes and their significance. Before starting our topic, we should know about enzymes. Enzymes increases the rate of chemical reaction by lowering the free energy barrier that separates the reactant and product. General properties associated with enzymes. Enzymes differ from ordinary chemical as a catalyst, which are having higher reaction rates up to the 10 raised to 6 to 10 raised to 12, milder reaction conditions, that is temperature, pH, greater reaction specificity, that is no side products, and capacity for regulation. Definition: catalyst. Catalyst, that is catalyzes the reaction but is not itself consumed during the process. Coenzyme classified as these are the part of active site of enzyme. They supply a reactive groups that are not available on the side chains of the amino acids. Coenzymes that can be synthesized from common metabolites are referred to as metabolite coenzymes. Those that cannot be synthesized are derived from the vitamins are known as vitamin derived coenzymes. Coenzymes can also be further classified based on whether they remain bound to an enzyme at all times or not. Many come on and off enzymes carrying electrons, etc., from one enzyme to another, and these are referred to as co-substrates. That is, co-substrates are actually the substrates in the reactions. They are altered in the course of reaction and dissociate from active sites. The original co-substrate structure has to be regenerated in the subsequent independent reaction catalyzed by another enzyme, and thus co-substrates are continuously recycled in a cell, whereas the actual substrate typically undergo further transformations. Coenzymes that remain bound either covalently or non-covalently via a numerous weak interactions to an enzyme are known as prosthetic groups. Prosthetic coenzymes must return to their original form during each catalytic cycle, and most of the coenzymes are derivatives of vitamins. In the fact, the biochemical functions of vitamins are exerted through their respective coenzymes. Let's look for the vitamins and their coenzymes. As in a chart, the thymine, the vitamin which is having the coenzyme thymine pyrophosphate, riboflavin which is having flavin mononucleotides, niacin that is a nicotinamide adenine dinucleotide, pyridoxine which is having the coenzyme pyridoxal phosphate, pantothenic acid which is having the coenzyme A, while folic acid which is having tetrahydrofolate, biotin that is biocytin, while cyanocobalamin which is having the coenzyme that is methylcobalamin, deoxyadenosylcobalamin. Coenzyme vitamins and direct precursors of coenzyme vitamins are found in foods in relatively small but significant amounts. In order to create a supplements with the larger amounts of vitamins, manufacturers have synthesized vitamins that are similar to but not the same as coenzyme vitamins that are needed to facilitate a metabolism. Let's begin with metabolite type of coenzymes. Examples of metabolite coenzymes are ATP, adenosine triphosphate, and nucleotide sugars such as uridine diphosphate glucose, that is UDP glucose. ATP has a central function unit, that is phosphoryl group transfers as occurs in reaction catalyzed by kinases. Pyrophosphoryl and adenyl AMP groups also can be transferred from ATP to the substrates, and nucleotide sugar coenzymes such as UDP glucose are involved in carbohydrate metabolism. UDP glucose is a carrier of glucose units used in glycogen synthesis, whereas ADP glucose is a carrier of glucose units used in starch synthesis. Vitamin derived coenzymes and nutrition. Vitamins are required for synthesis of vitamin derived coenzymes. Thus, vitamins must be obtained daily from the sources which are able to synthesize them, such as fruits and vegetables. Nutritional deficiency diseases results when daily consumption of vitamins is inadequate. Food supplementation programs. They still are common in many third world and developing countries. Vitamins can be classified into the water soluble, that is B vitamin and vitamin C, while fat soluble, that is vitamin A, vitamin D, vitamin E, vitamin K groups. The consumption of excess water soluble vitamins simply results in their excretion in the urine, and consumption of excess fat soluble vitamins can lead to the toxic conditions known as hypervitaminosis. Now let's look one by one vitamins and their coenzymes NAD plus, NADP plus. These compounds were the first coenzymes to be recognized, and the nicotinamide coenzymes are nicotinamide adenine dinucleotide NAD plus and nicotinamide adenine dinucleotide phosphate, that is NADP plus. Both are derived from the vitamin niacin, that is nicotinic acid, and the deficiency of which causes the disease pellagra. Niacin is present in many cereals, meat, and legumes, and humans can take the small amount of niacin from amino acid tryptophan, but not enough to meet our requirements. And these coenzymes also are commonly called pyridine nucleotides due to the resemblance of niacin ring to the organic compound pyridine, and the structures of oxidized NAD plus and NADP plus and reduced form of that is NADH and NADPH forms of pyridine nucleotides are shown in each molecule contains two nucleotides which are the moieties linked by the phosphonoanhydride linkage, hence the dinucleotide designation. One of these nucleotides is adenosine monophosphate, that is AMP, which is a common component of many coenzymes. The NADP plus and NADPH forms contain a phosphate attached to two hydroxyl groups of AMP group. The phosphate is important in enzyme recognition, namely NAD plus is used only in oxidative reactions of catabolism, whereas NADPH is used in the reductive biosynthesis reactions of anabolism. The pyridine nucleotides functions as electron carriers in the oxidation reduction reactions. In general, NAD plus is used in reactions where an alcohol group is converted to a ketone or aldehyde group, and the oxidized forms pick up the electrons from substrate in the form of hydride ion H minus, which is carried at position 4 of the nicotinamide ring. The hydride can then be transferred to another acceptor, regenerating the oxidized form of pyridine nucleotide. And the participation of NAD plus in the metabolism is illustrated in the lactate dehydrogenase reaction during the reaction. A hydride is transferred from lactate carbon 2 to NAD plus, forming NADH and pyruvate. A proton also is removed from the lactate by histidine in the active site, and this proton is eventually released into the cytoplasm. Next example, that is FAD and FMN type. The flavin nucleotides, that is flavin adenine dinucleotide FAD and flavin mononucleotide, that is FMN, are derived from the riboflavin, that is vitamin B2. Vegetables, grains and meats such as liver are rich sources of riboflavin. Riboflavin itself is composed of isoalloxane ring and molecule of ribitol. A phosphate is attached to the ribitol in FMN, and the phosphate plus AMP is attached in the dinucleotide FAD. The riboflavin and the flavin nucleotide are colored bright yellow because the isoalloxane ring absorbs the light strongly, in contrast to NAD plus and NADP+. FMN and FAD are the prosthetic groups which remain tightly bound to their enzymes and do not subtle back forth between the enzymes. Enzymes that contain these coenzymes are called as flavoenzymes or flavoproteins. FMN and FAD also carry the electrons in the oxidation reduction reactions. They are more powerful oxidants than NAD and NAD+, and often participates in the reactions where the carbon-carbon single bond is oxidized to double bond, and flavin nucleotides can accept and donate electrons either one or two at the time. The fully reduced forms that carry the two electrons are FMNH2 and FADH2. The partially reduced forms that carry one electron are relatively stable free radicals called semiquinones. Let's take another example, that is coenzyme A. Coenzyme A is an important coenzyme that is used to carry the acyl groups in the numerous reactions of carbohydrate and lipid metabolism. Coenzyme A, that is abbreviated coenzyme, and HS-CoA is composed of 2-mercaptoethylamine, which contains a free SH group, the vitamin pantothenate, that is vitamin B3, and the molecule ADP, in which the ribose 3-hydroxyl is phosphorylated. Acyl groups are carried in thioester linkage by SH group of 2-mercaptoethylamine, and these compounds are referred to as acyl CoAs. While next category, that is vitamin biotin. Biotin is the prosthetic coenzyme that catalyzes the carboxyl group transfer and ATP dependent carboxylation reactions. It is covalently linked to the active site of its host enzyme by the amide bond to E amino group of lysine residue. Biotin deficiency is rare in humans since very small quantities are required, and since it is synthesized by intestinal bacteria. Do not eat raw eggs. Next category, that is the fat soluble vitamins. All fat soluble vitamins are poorly soluble in water due to the presence of large nonpolar regions. They are absorbed in the intestines along with the other dietary lipids and transported in the blood by specific carrier proteins or plasma lipoproteins. That is vitamin A, retinol. Vitamin A is 20 carbon lipid that can be derived by cleavage of beta carotene present in vegetables. It also prevalent in sources such as the liver, egg yolk, and milk products. Vitamin A contains a alcohol group and is known as retinol. The retinol and retinoic acid forms are very important regulators of gene expression, particularly during the development. A deficiency of vitamin A causes a night blindness. Vitamin D3, that is cholecalciferol, and D2. Vitamin D is the name for the group that is related with the lipids that are precursors of hormone 125 dihydroxycholecalciferol, which is very important in calcium metabolism. While vitamin D3, that is the cholecalciferol, is formed in the skin in the people who get the significant exposure to the sunlight by photoreaction, in which a precursor of cholesterol is converted to the vitamin, and cholecalciferol subsequently is converted via two hydroxylation steps to 125 dihydroxycholecalciferol, the active form. Let's look towards next vitamin, that is vitamin E, a tocopherol. Vitamin E is an important lipid soluble antioxidant that helps in protecting polyunsaturated fatty acids in the membrane phospholipids from oxidative damage. Vitamin E, also known as a tocopherol, that contains a phenol group that can carry an electron as a stable free radical. Vitamin E probably picks up the electron from the lipid free radical species and pinching prolongation of free radical damage. Interestingly, the free radical form of vitamin E is recycled to its original state by the reaction with the vitamin C, which also plays an important role in protecting against oxidative damage. The reduced form of vitamin C is eventually excreted in the urine. Vitamin E is a prevalent in fish oils. Now let's move towards next vitamin, that is vitamin K, phyloquinone. Vitamin K serve as coenzyme in an important post-transitional modification of some blood coagulation proteins. In this reaction, a glutamate residue are carboxylated to their carboxyglutamate form, which binds the calcium. These divalent cations serve as a bridge by which coagulation proteins are anchored to platelets, which home in to the area where the clotting is needed. Vitamin K reversibly carries an electron in its quinone ring that is used in the carboxylation reaction. A number of medically important drugs blocks the recycling of vitamin K carries and decrease coagulation protein carboxylation, thus reducing clotting in patients suffering from clotting disorders. One of the original vitamin K analog developed warfarin also is used as a rat poison. Now friends, this is all about vitamin and their coenzymes. Now let's begin with the module 2, that is metals as a cofactors. Many enzymes use as a metal ions as a cofactors. Carboxypeptidase A is one such an enzyme requiring a zinc ion for the catalytic activity. Carboxypeptidase A exploits the chemistry of its zinc ion, which is a positively charged, in order to increase the rate of the reaction. In which, first, explain the role of zinc ion in the first step of catalysis, the Michaelis complex. Second one, that is explain the role of zinc ion in the second step of reaction, the tetrahedral intermediate. Many enzymes require a metal cations for the activity. Metal activated enzymes require or are stimulated by cations such as potassium, that is K plus, calcium, that is Ca2 plus, or magnesium, that is Mg2 plus. Often the metal ions is not tightly bound and may even be carried into the active site attached to the substrate, as occurs in the case of kinases, whose actual substrate is magnesium ATP complex. In metalloenzymes, the metal ion is tightly bound and participates in catalytic mechanism more directly. A good example is zinc ion in the active sites of carbonic anhydrase, where the zinc promotes ionization of water molecules in the active site to form a hydroxyl group. Formation of hydroxyl group is favored because it is stabilized by electrostatic interaction with the zinc. The hydroxyl group subsequently attacks CO2 molecule that has entered the active site, yielding the bicarbonate as a final product of this reaction. In other metalloenzymes, metal ions participate directly in electron transfers during the oxidation reduction reaction. This is all about a metal as a cofactor. Now friends, let's move towards the conclusion. With all these information, here we come to the end of the topic. In the important unit, enzyme 3, which is an interesting unit of the subject biochemistry, we discussed the concept of coenzyme and vitamin as a coenzymes and their significance, and also the concept of metal as a cofactor. So dear students, don't forget to review all of them in your self-study. If you have missed any of the module or content, you may log on to our website, that is www.cec.nic.in, to download videos, FAQs, LOR and other contents so that you can stay updated with this chapter. Keep studying. Good luck. Goodbye.